Okay. Now, early some scientists made attempts to classify. Okay, but scientific criteria were not used. Okay. Scientific criteria were not used. Okay, so what criteria they use? They use criteria like food, shelter, and clothing. The first time scientific attempt was made by Aristotle. Scientific attempt by Aristotle. So he has divided plants and animals. Okay. So plants he has divided on the basis of morphological features. Morphological features. Okay. On the basis of habit. Okay. How? Like herbs, stem, soft, flexible, which are less than 2 meters. Okay. Then shrubs, woody, which are medium in size. Okay. And then trees. Trees means which are thick woody plant but taller trees. Okay. Now animals. He has divided animals on the basis of RBC. OTB means on the basis of RBC. Okay. Anima, organisms in which RBCs are present, they are called anima. And in which RBCs are absent, they are called anima. A means absent. Okay. In which RBCs are absent. So first scientific attempt was made by Aristotle. So this is first kingdom of classification. Okay. Next, two kingdom classifications. Two kingdom classification. Two kingdom classification was given by Linnaeus. Okay. Kingdom, that is plant T. Kingdom word, so AE suffix. Okay. Animalia, IA suffix. Whenever I am telling, whenever we will write kingdom, so we will not write plant or animal. We will write planty and animalia. Okay. Now, what are the bases? Let us write some points. Suppose cell wall. Cell wall is present in case of animals, plants, absent in animals. Then the second factor, locomotion. In plants, locomotion is absent, whereas in animal, locomotion is present. Mode of nutrition, autotrophic for plants, heterotrophic for animals. Now next, response to stimuli. Poorly developed in plants, well developed in animals. Last one is contractile vacuum. Contractile vacuum. Absent in plants, present in animals. Let us take some examples. Examples which was given by Linnaeus for animal. Okay. Protozoans, invertebrates, vertebrates. Now in plant, what he has done, in plants, he has placed bacteria, fungi, algae, bryophyte, pteridophytes, gymnosperm, as well as angiosperm. All these things he has placed in plant kingdom. So you can see that you can say that this is a demerit of this two kingdom classification. So this is what about two kingdom classification. So let us see its drawback. What is the drawback of this two kingdom classification? What is the drawback of this two kingdom classification? What is the drawback of this two kingdom classification? Now, number one, first point, differentiation of prokaryote and eukaryote was absent. Next, differentiation of unicellular and multicellular organism was absent. Differentiation of autotroph and heterotrophic organism is absent. And one more disadvantage, some organisms was there, they have a feature of both plants and animals. Means they are dual organisms. We have seen that prokaryote and eukaryote, like bacteria as well as angiosperm. Bacteria is a prokaryote, angiosperm is a eukaryote. Both differentiation absent. Why? Because both are placed in plant kingdom. Unicellular, multicellular. Unicellular is bacteria. Multicellular, unicellular bacteria, multicellular gymnosperm, angiosperm, both are placed under plant kingdom. Then autotrophic, heterotrophic. Okay. Green plants are autotrophic, but fungus are heterotrophic. So differentiation of autotrophs and heterotrophs placed under differentiation was absent. And some organisms were there who have both plants and animal feature. Where you will put those organisms? So these are the drawbacks of this two kingdom classification. Next, I'll give you some example where you can find in one organism these two features, like dual organism. See the examples. Chlamydomonas. Okay, they possess cell wall. They are autotrophs. Means this is a feature of plants. Next, they also locomote and they have contractile vacuum. They have contractile vacuum. Means this is the animal feature. Next, dual organism Euglena. Here, cell wall absent. And cell wall absent means again animal feature. Autotrophs, plant feature. That's why this drawback led to the evolution of three kingdom classification. Okay. So first two kingdom means plant and animal was there. One additional kingdom was added. Okay. Three kingdom classification. Now, this three kingdom classification term was given by Haeckel. Now, which is the third kingdom? Third kingdom is Protista. Okay. In Protista, what happened? Morphological complexity absent. Morphological complexity and tissue level differentiation. Tissue level differentiation. Both are absent. So what he has done, unicellular eukaryotes. So what organisms are placed under Protista? Number one, unicellular eukaryotes like protozoa, slime molds. Next, multicellular eukaryote, fungi, algae, and then prokaryotes like bacteria. All these organisms are placed under Protista. Okay. So this is about three kingdom classification. First, three kingdom classification is Plantae, Animalia, and Protista. Okay. Now again here one drawback: morphological complexity and tissue level differentiation absent. So next is four kingdom classification. Four kingdom classification. Now in four kingdom classification, one additional kingdom again here added, that is Monera. In Monera they have placed prokaryotic bacteria. Okay. Now five kingdom classification. This is very important, student. Five kingdom. This is very important, and its scientist name is very important. Various times they have asked question with this. Whitaker. R.H. Whitaker. Very important. Four kingdom. Now what we have till now? We have Monera, Protista. Third kingdom, fungi. This one newly added. Fourth one, Plantae. Fifth one, Animalia. This is the five kingdom classification. Okay. Monera, Protista, Fungi, Plantae, Animalia. Okay. Now, what are the features of fungi? Features of fungi. Features of classification. Features of five kingdom. Now, he has divided prokaryote and eukaryote on the basis of complexity of cell structure. Complexity of cell structure. He has divided into prokaryotes and eukaryotes. Okay. Next, complexity of body organization. Okay. On the basis of that, he has classified unicellular and multicellular organisms. On the basis of mode of nutrition, he has classified into autotrophs and heterotrophs. In heterotrophs, three organisms are there: parasitic, saprophytic, holozoic. This was the main criteria by Whitaker. Fourth one is reproduction. Fifth one is phylogenetic analysis. What is the meaning of phylogenetic analysis? Means suppose you are comparing two organisms on the basis of evolution. They are called phylogenetic analysis. Means evolutionary relationship of organism. Okay. So these are the main five basic features of five kingdom classification. So let me tell you all the features in a comparative manner. Let us see the features in a comparative way. Okay. Monera, Protista, Fungi, Plantae, Animalia. So, first basis cell type, second one cell wall, third one nuclear membrane, fourth one body organization. So cell type means Monera are prokaryote, Protista eukaryote, Fungi eukaryote, Plantae eukaryote, Animalia eukaryote. Cell wall. In Monera, cell wall is present, but they are non-cellulosic. Okay, non-cellulosic. Non-cellulosic means they are not made up of cellulose. Some proteins it is present. In fungi, cell wall is also present, but they are also non-cellulosic. In fungi, cell wall is made up of chitin. In Plantae, it is made up of cellulose. And in animals, cell wall is absent. Nuclear membrane absent in Monera. And rest all kingdom, nuclear membrane is present. Nuclear membrane is present. Body organization, they are unicellular, multicellular. But loose tissue, you will get here loose tissue. This tissue are not original student. Okay. True tissue formation is absent. Okay. True tissue formation is absent. Okay. This is about fungi. Next, what about in planting? Tissue formation as well as organ formation through planting. What about in animalia? Tissue formation as well as organ formation as well as organ system formation. These are the four comparative study of Monera, Protista, Fungi and Plantae and Animalia. One more feature is there on the basis of mode of nutrition. Let us see, students. One more feature. Okay. See, one more feature. Like, let us see on the basis of mode of nutrition. See, Monera are autotrophs as well as heterotrophs. Okay. Now, in autotrophs, they are photoautotrophs as well as they are chemoautotrophs. Photoautotrophs means they synthesize food with the help of light. Chemoautotroph means they synthesize food with the help of chemicals. And in heterotrophs, they are parasitic, saprozoic. In protists, they are also autotrophs as well as heterotrophs. Now in heterotrophs again, parasitic and saprozoic, same. In fungi again, same, Heterotrophs. Heterotrophs again same parasitic. Suppose I am denoting it by P and denoting it by S. So P S. Here also P S. What about Fronti? They are only photoautotrophs. They are also autotrophs, but they are photoautotrophs. And Animalia, they are heterotrophs. Again, parasitic. In Animalia, we have seen holozoic also. So this is 5 kingdom classification. Okay. But here also we will get one drawback. One or two drawback are there of this classification. That's why till now the most advanced classification is 6 kingdom classification, which was given by Carl Lewis. Okay. Till this, any doubt, any one of you? Students? Students, any doubt? Yes? Tell me. Anyone? Mode of nutrition फिर से एक बार बोल दे. Okay, fine. Now suppose let us take mode of nutrition. First kingdom, Monera. Okay. They are autotrophs. They can synthesize their own food as well as they are heterotrophs. In autotrophs, those need light, they are called photoautotrophs. Photoautotrophs. And those can synthesize with the help of chemical, they are called chemoautotrophs. And in heterotrophs, some are parasitic and some are saprozoic. Saprozoic means those live on dead and decay organism. If I take for Protista, they are autotroph, but they are only photoautotroph. They are not chemoautotroph. What about heterotroph? Same, parasitic and saprozoic. Okay. If you take for fungi, in fungi, autotrophs absent, only heterotrophs. Clear with these 3 kingdom? Clear with this 3 kingdom? Fourth one is plantae. Wait a minute. Wait a minute. Huh? Pen is not open. Now, next one is for plantae. They are only autotrophs. Which kind of autotroph? They are photoautotroph. They are photoautotrophs. What about Animalia? What about Animalia? They are heterotrophs. Is it clear? Mode of nutrition is clear to all of you? Students?
Yes, ma'am.
Clear now? Now let us see the drawback of this kingdom. Drawback. First drawback, algae. First drawback is algae. Okay, now. Now in algae, we have put Monera. That is example of blue-green algae. We have put in Monera, kingdom Monera. Okay, now. Now, blue-green algae, he has put in Monera. Okay. Then, in Protista, he has put dinoflagellates. Okay. And in Plantae, that is kingdom Plantae, he has put green algae, red algae, and brown algae. You know. The second one, he has not defined any status of viruses. Status of viruses were not established. Were not established. So this is the drawback of five kingdom classification. Now next one is six kingdom classification. In six kingdom classification was given by Carl Woos. Six kingdom is given by Carl Woos. So he has divided Monera into two kingdoms. One is Archaebacteria and another is Eubacteria. Another is Eubacteria. Okay. On what basis he has classified this Monera? On what basis? Okay. He study on the basis of study of sequence of 16S ribosomal RNA gene. On this basis, he has distinguished this Archaebacteria and Eubacteria. Now, the cell wall of Archaebacteria, is made up of pseudomurine. Whereas for Eubacteria, it is made up of 50-doglycan. peptidoglycan or purine. Okay. Now, if I talk about the cell membrane, what you will see here, suppose this is a fatty acid. This is a fatty acid. This is a fatty acid. Okay. Again, this fatty acid, but they are actually attached. Okay. They are attached like this. They are attached. So we can say that these are branched. Fatty acids are branched. What about in eubacteria? This is fatty acid. This is fatty acid. This is fatty acid. This one. This one. This one. Now this is one layer of fatty acid. This is another layer of fatty acid. So we can say that the cell membrane of eubacteria consists of lipid bilayer. Bi means two. You can see two layer of lipid bilayer. Fatty acid here, unbranched fatty acid. Unbranched fatty acid. So suppose this is a fatty acid. Okay, now this is the head portion, this is the tail portion. The head portion is hydrophilic. Okay, head portion is hydrophilic. And tail portion is, this is tail of fatty acid. Tail is hydrophobic. Phobia means hate. Hydro means water, means they hate water. Hydro means love. Philic means love. Hydro means water, means they love water. Means head, they love water. Tail, they hate water. So this is the condition of cell membrane and cell wall in archaebacteria and eubacteria. Till this clear? Clear to all of you? Any doubt, ask me. Any doubt, students?
Cell membrane.
Okay. Cell membrane of archaebacteria. Okay. This is a fatty acid. Fatty acid. Fatty acid. Now these fatty acids are actually branched. You can see the structure. These fatty acids are branched. Means they consist of branched fatty acid. Okay. Now what about eubacteria? In eubacteria, we'll see this is fatty acid, fatty acid, fatty acid, fatty acid, fatty acid. So it is actually unbranched fatty acid. So, unbranched fatty acid. Clear with this? Now this is one layer of lipid. This is another layer of lipid. That is why this type of structure is called lipid bilayer. Lipid bilayer. Clear with this?
Yes, ma'am.
Yeah. Now, as I told you, the basis is 16S ribosomal RNA. So, what is this actually 16S ribosomal RNA? Suppose DNA is the genetic material. This DNA will undergo transcription of RNA. This DNA will undergo transcription to form RNA. This RNA will undergo translation to form protein. Okay. Now, this protein actually ribosomal RNA as well as they form protein. Now, in ribosome consists of two subunits. One is 50S, another is 30S. In this 30S subunit, students, this 16S ribosomal RNA is present. 16S ribosomal RNA is present. Okay. Seeing this 16S ribosomal RNA, he has isolated Archaebacteria and Bacteria. Clear. So he gave three domains of life. Okay. What are the three domains of life? What he has given? Here I will to say Carl Us. Okay. Carl Us. Okay. So he gave three domains of life. He gave three domains of life. What are the three domains? First one is Archae. This is one domain. In this domain, what are the kingdom? Archaebacteria kingdom. Second domain is bacteria. Bacteria. Second domain is bacteria. In this Eukaryotes kingdom. Eukaryotes kingdom. And third domain is Eukarya. Third domain is Eukarya. In this, you will get Protista, Fungi, Plantae, and Animalia. All these kingdom. He believed that all archaeodomains, that means all these domains. He believed that all archaeodomains, he believed that all archaeodomains who have evolved from a common ancestor called progeny. What is the name of the ancestor? That is progeny. Okay, progeny. So I'll start with kingdom Monida. Okay. Till this, any doubt? Any one of you? Till this, any doubt? Any one of you? Till it, any doubt? Any one of you? Any doubt? Any one of you? Our first kingdom, kingdom Monida. Kingdom Monida. It includes all prokaryotes. The prokaryotes are always unicellular. They are found everywhere, like soil, air, water, in extreme habitats, like hot water spring, desert, snow, deep ocean. That's why we are using the word cosmopolitan for Munira. Cosmopolitan means they are found everywhere. Okay. They are the simplest, smallest, and they are most abundant organism. Most abundant organism. They are first inhabitants of Earth. They are first inhabitants of Earth. And bacteria are the sole member. Bacteria are the sole member of this kingdom. Kingdom Monida. Okay. So now next one, it includes bacteria, archibacteria, cyanobacteria, cyanobacteria that is blue green algae, mycoplasma, rickettsia, actinomycetes, and chlamydomonas. One by one, we read all these things. These are included in Monida. Okay. Now, next thing what we'll see, that is the characteristics of the Monida. First characteristics which we will deal here, that is for nutrition. Okay. Characteristics, first characteristics, that is nutrition. Okay. They are autotrophs, as I told you earlier. Two kind of autotrophs, photoautotrophs and chemoautotrophs. As I told you earlier, students, just a quick revision of that. Heterotrophs, parasitic, symbiotic, and saprophytic. Now, this saprophytic mode of nutrition is the main mode of nutrition. Is the main mode of nutrition. Okay. And second one is reproduction. Now, their main mode of reproduction is asexual reproduction. Why? Because true sexual organ is absent. True sexual organ is absent. Clear with this, students? Clear with this, all of you? Clear with this?
Yes, ma'am.
Okay, fine. So we have done with the today's class. Okay. Thank you all.